continue. And first up, we'll have an acknowledgement of country. So in the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and the connections to land, sea and community. We pay our respect to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past, present and emerging, and extend a welcome to any Aboriginal or Torres Strait Islander peoples on this call today. So today's theme is around identifying and targeting disrupted systems in MND. We have Dr. Alison Hogan from Macquarie University, and she's a current Beryl Bailey Fellow. And her work sort of delves into looking at these disrupted systems and trying to work out what's actually going on with them. And then we also have Dr. Jeremy Lum, who is the current Bill Gold Fellow, and he's going to tell us more about how he's been trying to identify potential therapies and targets that may have come from looking at these disrupted systems. So as previously, each presenter is going to talk for about 20 minutes, and then at the end we'll have a combined Q&A session at the end of the session. So please submit your questions through the chat or Q&A function in Zoom, or if you're on Facebook, then fire in your questions through the comments, and we'll collate all the questions and try and get them asked at the end. So that's it. That's all you need to hear from me. So I'll stop sharing my screen. And Alison, it's all yours. Take it away.
Thank you.
That's all good.
All good? Okay.
Yep.
So good evening, everyone. As Kristen said, my name's Alison. I work at Macquarie, and I was fortunate enough to be awarded the Beryl Bailey Fellowship last year. So tonight I'm going to tell you about some of the work that sort of led up to that fellowship, and then where we're planning on taking it thanks to MND Australia. So I thought this audience is probably fairly well aware of what MND is, sort of what it looks like clinically, but what you may be less aware of is just what a complex disease it is. Around 10% of our patients have a known family history of the disease, so they have clear genetic links causing MND. Now, there are some diseases like Huntington's disease, for example, that have a single gene that carries mutations, and these mutations cause the disease. MND, in those cases, looking at the function of that gene can give us a really clear insight into what systems or what biological pathways are damaged, and therefore how disease develops. MND is not that straightforward, unfortunately. Mutations so far have been identified in around 43 different genes, each of which have a different function. On top of that, we have around 90% of our patients that don't actually have a known family history of the disease. So MND in these patients is believed to develop due to a combination of genetic mutations that we may not be aware of yet, and also environmental factors. So the great challenge that we face is really trying to figure out how all these different genetic mutations, different environmental factors all lead to this same outcome, which is the death of the motor neurons. Now, a huge amount of work has been done looking at the function of these 43 genes and developing models that are based on some of the mutations. And this work has led to the identification of quite a long list of systems or cellular pathways that are dysregulated and contributing to motor neuron disease. As you can see, this list is quite long and it's quite varied. I'm not going to go into detail about everything there, but as you can see, we've got damage at the DNA, at the RNA, at the protein level. We've got problems with the movement of proteins around motor neurons. There is disruption to the pathways that produce energy in the cell, so resulting in excessive amounts of stress. The systems that are supposed to help deal with that stress are also dysregulated. We know that motor neurons are sort of overexcitable in MND. And we also know that motor neurons are not the only cells that are involved in progression of MND. Particularly cells of the inflammatory response have been implicated. So how can we develop treatments for such a wildly complex and varied disease? There are some really exciting therapies that are being developed that target sort of specific genes and genetic mutations. And Jeremy, who's going to be speaking after me, is going to talk to you about some of those. However, what we're still really working on is developing treatments that are going to be effective as well for these 90% of patients that don't have a known genetic cause of their disease. So in order to do this, we need to gain a really clear understanding of each of these systems, how they're disrupted in MND, and how this disruption contributes to motor neuron death. And if we can do that, we can identify targets within these systems. Targets that are most important, contribute most to disease progression, and then we can figure out how to modify them with drug therapies. So one way to approach this is we can look in specific detail at one particular pathway. We're taking a slightly different approach, which is that we're looking at one particular protein, and this is our protein called SFPQ. We're particularly excited about SFPQ because evidence we have so far suggests that it is changed, its behavior is changed in patients, regardless of whether they have a genetic mutation or if they're one of these 90% of sporadic patients. And it's really rare to find something that is common to the vast majority of patients in MND. There's really only one other protein that's been shown to consistently act differently across the vast majority of patients, and that's TDP43, which I get anyone that's sort of tuned into these previously is probably familiar with. So changes in TDP pathology, changes in TDP protein behavior were identified roughly 12 years ago, and a huge amount of work has been done looking at the causes and consequences of these changes, and that's given us a huge amount of insight. We think SFPQ is equally exciting and can have a similar impact. Now, one bonus of studying this protein SFPQ is that it functions in a number of these different pathways or systems that we know are disrupted. So it gives us a chance to look at multiple systems at the same time and also how they interact with each other. So before I go into more detail about SFPQ, I do need to give you a very quick biology tutorial. It will be very quick. So basically, this is the structure of our motor neurons. They have a nucleus, which contains the DNA, and the rest of the cells made up of the cytoplasm. And that extends all the way down the length of the axon, where it connects with the muscle and tells the muscle when to contract. So it's slightly more complicated, but not too much. The other thing you need to know is how proteins are made in our body. So we have DNA, which, as I say, is in the cell nucleus. It's essentially just two strands wound around each other. So the cell makes a copy of one of these strands, and this is your RNA. At this stage, the RNA contains sections called introns. The next step of protein production is actually removal of these introns. They're literally chopped out, and the bits of RNA that are left behind fuse together to form a much shorter, sort of mature RNA, and that mature RNA just forms the template for how your protein is made. So biology lesson over, back to SFPQ. I guess to start with, why do we get interested in this protein in the first place? And the answer is that sort of over the last 5 years or so, a number of studies have come out looking at different models of MND. And all of these models have shown that SFPQ is behaving differently compared to how it normally would. As you can see, these models were in the pig, a couple of mouse models, and also a couple of different cell models. One of them is just sort of standard cell lines. The other are stem cells. So these are developed from patient tissue. So you take a skin biopsy is the most common way, and then you can reprogram those cells to form these stem cells. So these are a fantastic model. What really got our attention from all these studies was the fact that all these different models are based on different genetic mutations. So I've shown the mutation in the brackets after each one, just to give you an idea of the variety. So these are changes that they're reporting across different genetic subtypes of MND. And not only that, but they've also shown that these changes are present in sporadic patients. So those 90% of patients that don't have a clear genetic link to MND. So this is why we got interested in SFPQ. But before we got too excited, we wanted to make sure that these findings from models are actually relevant to patients, whether they're actually something that we can see in patients. So to investigate this, we did a big study where we examined SFPQ behavior in as many patients as we could, and patients across a range of with different genetic mutations, and also from that large sporadic group. And I'll just take you through the sort of two key findings from that study now. The first of these was we found differences in where SFPQ is actually located within the cell. So in a healthy motor neuron, SFPQ is mainly found in the cytoplasm, in the nucleus of the cell. So here we have an image of human motor neuron. The grey channel, it just shows it's a neuronal marker. So we can see the outline of the nerve, and we can also see where the nucleus sits within there. To make it clearer, I've added the yellow dashes around the nucleus. The red channel is just an antibody which stains SFPQ, so it tells us where it is within the cell. So when you look at the merged image, hopefully you can appreciate that SFPQ is really packed into that nucleus. There's a lot of it in there and not so much sort of floating around the cytoplasm. Now, what the animal and cell models, and what we also saw in patients, was that in MND, this SFPQ protein moves from the nucleus out into the cytoplasm of the cell. So we lose nuclear expression and probably nuclear function. And what we also see in patients, which wasn't reported in the models, was that as well as moving into the cytoplasm, SFPQ also forms these really large clumps or aggregates within the cell. And this is something we saw in both our familial and sporadic patients that we were examining. So the second change in protein behavior we saw was actually how the proteins are made. This comes back to the biology lesson before. Hopefully you'll recall that the introns are usually removed when the RNA is processed. In MND patients, we found this is changed for SFPQ. Instead of cutting out all of the introns, we often get retention of one particular intron in our final RNA product. And we sort of hypothesized that this would lead to a protein that acts quite differently. So we had a look at this. Again, we saw this in familial and sporadic patients. But we had a look at what this intron retention does to the protein behavior in cell culture. So here we've just put either the wild type or normal SFPQ or SFPQ that carries that intron still into our cells, and then let the cells produce whatever protein sort of comes from that. Here, blue channel, we have a nuclear marker. So it just tells us where the nucleus is in the cell. And this time SFPQ is green. Hopefully you can appreciate with our wild type or normal SFPQ, those cells that are green, there's a perfect overlap between SFPQ and our nuclear marker. So again, as we expect, SFPQ is really mainly in that nucleus. Whereas when we have that intron retained, the protein behaves really differently. You can see a lot more of it spread out throughout the cytoplasm. And this happens really quickly. We see this within about 24 hours. And about 48 hours after that, we start to see that the protein's forming these, again, really big clumps in the cytoplasm. And if you look at the merged image, you can appreciate that these clumps are outside the nucleus. So these findings, as I say, we saw the nuclear mislocalization, we saw the aggregation, and we saw that change in intron retention in both familial and sporadic patients. So this really convinced us that SFPQ is an exciting avenue, exciting way to investigate systems in MND. So we're now moving on to what I find the really exciting part, which is figuring out what these changes do. So, what causes these changes to start with? So what's happening upstream to cause SFPQ to act differently, and then downstream, what effect does this have? So basically, we're always coming back to our overall aim to figure out how we can prevent sort of these adverse effects from happening and stop disease progression. So this is where I'm going to change tack a little bit and tell you about where the project's going next, thanks to the fellowship. So for this stage of the project, we are going to move back into using models, at least initially, because models allow us to sort of change the genetics and change protein behavior. We're going to use a combination of cell culture models and also living animal models. Cell culture models are fantastic. I've shown you sort of a couple of examples of that. They tell us a lot, but they are a very simple system. It's one cell in a very controlled environment in a dish. The central nervous system is obviously, or the nervous system is obviously very complicated with a lot of cells and a constantly changing environment. So to really sort of get reliable results, we do need to look in a living organism. And at Macquarie, we're really fortunate that we have an amazing zebrafish facility. Now, some of you listening may already be familiar with zebrafish and their use as models of neurodegenerative disease, but I imagine not everyone is. So I'll just take a couple of minutes here to sort of convert anyone who's questioning sort of why we would do this or how relevant our findings will be. Starting off, zebrafish are like us, they're vertebrates. So on the evolutionary scale, they're surprisingly close to humans. And this means that they share a lot of the same genes and the same proteins that we do. And this includes the genes and proteins that have been implicated in MND, including SFPQ and TDP and other genetic mutations. In addition to this, if we just look at the nervous system, zebrafish have very similar, essentially the same nervous system as we do. They have the same cells that interact in the same way, functions in the same way, and it's the same basic structure. So this is why we say that it is relevant to human diseases. If we compare zebrafish to other vertebrate models that are often used, so usually mice and rats are probably the most common, advantages of zebrafish over these models: first of all, they lay eggs, so they develop externally to the body. And this means that sort of manipulating or changing their genetics is surprisingly straightforward. They also develop extraordinarily rapidly. From laying the egg, which is a single cell stage, within 2 days, they've developed, they have a developed central nervous system, they're moving freely, and we can start studying. So this really speeds up a lot of our studies. But my absolute favorite part of zebrafish is the fact that they're transparent when they're young. And this means we can do some really amazing imaging studies in a living animal, which just isn't possible in other vertebrate models. So to orientate you with the fish, this is just a schematic showing you where the head is, the tail, and the fact we've got the spinal cord running down the body of the fish. Here we have a fish that is about 2 days old. Again, head up this end, tail down that end. This fish is expressing a yellow fluorescent protein selectively within its motor neurons. So here we can actually see the spinal cord in yellow running down the length of the fish. And the little projections extending downwards from that are the axons leading out to the muscles. And we can get some incredible detail with these images. So this is one single motor neuron. This is the cell body in the spinal cord, and these are the axons extending out. And you can see that we can sort of label different structures in different colors. So again, we can get incredible detail and really beautiful images. So while those images are beautiful, the detail we can get from living zebrafish gets even more impressive. So in this video, we're looking at movement of SFPQ along the axon. So just a small segment of the axon in a living fish. So this fish is anesthetized, so it's not moving, but it's very much alive. And as you can see, SFPQ is moving up and down this axon at a fairly rapid rate. So I've got this here to, one, convince you how brilliant zebrafish are, but also to give you an example of one of the studies we're doing with SFPQ in the fish. Now, I mentioned it briefly in the introduction, but one of the systems that we know is disrupted in MND is the transport or the movement of proteins within the motor neuron. So proteins are produced in the body of the motor neuron, and they need to move all the way down the length of the axon to the endings. And this can be over 1 meter long. You have some cells that have the cell body in your spinal cord, and the axon goes all the way down to your big toe. So this transport system is essential. If it's disrupted, we know that the motor neuron dies. SFPQ is an essential component of this system. It connects the proteins that need to be moved to the proteins that are responsible for that movement. So I like to think of it as the proteins that need to be moved are the cargo. SFPQ is like the shipping container that holds the cargo. And then we have the motor proteins, which are the truck. So given the fact that we know SFPQ behavior is altered in MND and how essential this system is, we're looking at sort of what effect changes to SFPQ has in this system. Now, in the interest of time, I am running out of time. The last aspect of our planned zebrafish study I'll tell you about today gets us back to looking at multiple systems and how SFPQ contributes to their disruption. So here we have a fish that's expressing SFPQ, which has been tagged with a green fluorescent protein this time. And again, this expression is only in the motor neurons. So we can see the spinal cord fluorescing green, which is our SFPQ. And then we have, this is enlarged images of single motor neurons from different fish. And this is just to show you sort of how we can genetically manipulate our fish and change the behavior of the proteins. So first of all, we have just a wild type or normal SFPQ. So this is the body of the motor neuron. You can see that the SFPQ is mainly expressed in the nucleus, as we would expect, which is that inner purple circle. We've also got models where we have introduced a genetic mutation, which we think may be linked to MND in SFPQ. We can also change the behavior of SFPQ and make it leave the nucleus completely and just localize in the cytoplasm like we do see in patients. And we can also make it aggregate. So we can replicate all these pathologies that we do see in patients to see what effect they have. So we're still in the process of establishing these lines. But once we do, we will perform a big study where we will first obviously euthanize the fish, and then we will basically break them up so it's just a single cell solution. So all of the fish broken down into single cells. We then have a very fancy machine that allows us to pull out those cells that are fluorescing green. So these cells that are expressing our SFPQ, we can isolate those. We can then pull out the proteins that are within these motor neurons. And we can identify not only what the proteins are, but also how much of each of the proteins are present. And this is going to let us identify protein differences that develop due to changes in SFPQ behavior. So for example, what's different between normal SFPQ and SFPQ when it's forced out to the cytoplasm? And we have multiple software tools that will put all this information together, and that will give us an idea of what systems are being disrupted by this SFPQ pathology. Not only that, but it'll give us really detailed insight into exactly what proteins are changed. Some will be, expression levels will be increased, others will be decreased. And this will give us really clear indication of what's going on in each of these systems. Also, proteins that are changed the most, have the greatest effect, then become potential targets moving forward. So hopefully 12 months time, you'll have me back and I can give you an update on how this study is going, but I'll leave it there for tonight. Thank you very much for your attention. There's a lot of people that have contributed to this project so far ongoing. I'm very fortunate to work with two amazing teams at Macquarie, one led by Marco Morsch for the Zebrafish side of things, and Ian Blair for the genetics and cell biology side of things. Also quickly acknowledge the biobank at Macquarie, but also most importantly, patients and their families. Without sort of their donations of blood and other samples, the genetic studies and studies I've talked about today just are not possible. So that's it from me. Thank you very much for your attention. I will now hand over to Jeremy, who's going to talk to you essentially about the next step of the process. What happens when we have a target? Well, sorry, we've identified a target and want to see if we can modify it to improve disease progression. So over to you, Jeremy.
All right, thank you, Alison. So let me share my screen. Can you all see that? Yep. All right, cool. So thank you, Gethin and Laura, for inviting me to talk today. My name's Jeremy Lum, and I'm a postdoc research fellow at the University of Wollongong. And I'm a part of Professor Justin Yerbury's lab. So some of you may be familiar with Justin and his story with ALS. And for those of you interested, Justin's actually about to publish a book about his story and his family history with ALS, which is, I think, due to be published in mid-June. So keep an eye out for it. I think the proceeds are going to raise funds for MND research. So I don't think I need to talk too much about this because Alison's covered it quite well, but ALS is a type of motor neuron disease. And in our brain and spinal cord, we have motor neurons or nerve cells that are responsible for the movement of muscle. And obviously in a healthy motor neuron, it innervates that muscle and allows us to perform functions that we need to do throughout our lives. Unfortunately, in ALS, these motor neurons begin to die. And that leads to an inability to control those muscles that it innervates. And we see that the motor neuron dies along with the muscle that it innervates as well. And that's what causes paralysis. So we know as well that there is very limited treatments in both number and therapeutic efficacy for the treatment of ALS. So our lab is quite interested in developing these. So just a quick background, I think Alison's touched on the difference between familial and sporadic ALS. And I'm going to talk today about familial ALS in particular. So here on the y-axis, you can see that this is the number of genes that have been found to be associated with ALS. And then down here we have, I'll turn my pointer on. Down here we have a timeline. And you can see that the number of genes that have been associated with ALS has increased over time. And currently there are 43 genes that are associated with ALS. Now, in particular, we are quite interested in the SOD1 gene, which was the very first gene found to be associated with ALS back in 1994. Now, SOD1 mutations are responsible for about 20% of familial ALS cases. And in particular, there's been over 150 different SOD1 gene mutations associated with ALS, which is a huge number for one gene, because there's 20,000 genes roughly in the human genome. So what does that gene mutation mean? So again, I'm going to, I guess, cover a little bit of what Alison talked about, but in biology, but hopefully you'll pass the quiz at the end of the seminar. Here we have DNA, and this is kind of like a recipe book. It holds all the instructions that we need for our cells to be able to function and produce the things that they need. So what happens is we then have RNA, which is kind of like a bit of copying a little bit of this book, the instructions potentially to make a cake, say. And that template can then be turned into a protein. And in this case, it turns into a cake. And these proteins are vital for the cells to be able to perform their normal function. And when you have a SOD1 mutation, it's kind of like a typo within the recipe book. And that typo is carried through all the way to the protein level. And your protein, particularly for SOD1 mutations, turns out slightly different. And our lab is quite interested in looking at, well, what are the effects of this different form of SOD1? So I should say as well, this is a little bit about protein folding. So when the cell produces a protein, it doesn't come out like a fully folded protein ready to perform its function. It's a bit like origami. You start off with this blank piece of paper, and you then begin to start to fold it, and it becomes various intermediates before it's the fully formed, in this case, crane. So SOD1 goes down a similar pathway. It starts off very unstructured, or a blank piece of paper, and slowly moves down this pathway to an intermediate state or different intermediate states until it forms the final SOD1 protein to perform the function of the SOD1 protein. Now, when you're doing origami, if you make a mistake within this folding pathway, what ends up happening is you'll scrunch up your bit of paper and you'll toss it in the bin. Well, when we have gene mutations in SOD1 that are carried through to the protein, the cell does something similar, and it forms those protein aggregates that Alison talked about. So these protein aggregates are like big clumps of that SOD1 protein, and potentially other types of proteins as well. And when you have a gene mutation in SOD1, what ends up happening is you end up getting more of these protein aggregates. And one thing to point out here is that there is a more error rate in this folding pathway, particularly at the early stages. As the protein moves down and becomes more structured and folded, you get less chance that it will go into this protein aggregate. Now, when we look at tissue from patients with the familial SOD1 mutation, and we look at their spinal cord, you can start to see this is a motor neuron here, and this is actually a SOD1 aggregate. So it starts to fill the cell. And the cell actually can't get rid of these protein aggregates very well. And we think that these protein aggregates are what causes the motor neurons to start to die. So we're quite interested in looking at this, and to do this, we use a model. It's not a zebrafish model, but what we do is we use motor neurons that we can grow in the lab, or a motor neuron-like cell, and we can put into these cells either a healthy SOD1 gene or a SOD1 gene containing something associated with familial ALS. So we can grow these cells on a plate, and then we can look at them under the microscope. Now, we've put on them a fluorescent tag, and you can see here, this is the healthy normal SOD1 protein, and you can see it's quite well distributed throughout the cell. There doesn't seem to be any clumps, and we actually see that these motor neurons survive quite well with healthy SOD1. However, when we put in a familial SOD1 mutation, what we start to see are these big aggregates. So you can see it here outlined in blue, that start to form within the cell. And we've shown that this causes the cells to start to die. So we can look at the aggregate, and we can also look at the cell death, or the cell dying. So some work from Justin's lab over the past 10 to 15 years has really been focused on, well, what do these aggregates do to cause the cell to start to die? And we already saw that Alison showed that multiple systems are disrupted. So work from Justin's lab, particularly by Natalie Farrell, Dr. Luke McAlary, and Dr. Isabella Lambert-Smith, have shown that these aggregates stop the cell from making new proteins, which are vital to keep the cell functioning. It can disrupt the folding of these proteins, and it stops the removal of old proteins, which the cell needs to get rid of as it starts to age. It also interrupts energy production, cellular structure, and what Alison already touched on, protein trafficking, from getting proteins in this, I guess, what we would call a soma, so the cell body, all the way down to its neurites or synapses. And it also disrupts the communication of the motor neuron to innervate muscle cells. So we know that it disrupts all these systems. And then what that obviously leads to is the motor neuron dying. And that's what we're most interested in. We want to keep these motor neurons alive. Now, our efforts in our lab have focused on targeting all these systems, but we've started to really focus on, well, let's get to the root cause of the problem. Let's see if we can stop protein aggregation. So whether we can stop SOD1 forming these aggregates within the cell, which will hopefully, in the long run, stop all of these systems being disrupted. So one drug that we're particularly interested in, and some of you may be familiar, is the drug copper ATSM. Now, MNDRA were vital in funding the initial studies for copper ATSM in ALS. So it's a bit of a success story, I guess, because it's now in clinical trials. And what we did was we put copper ATSM on our cells containing the SOD1 mutation. Now, what you can see is that when we don't treat the cells with drugs, we get these bright puncta or bright little spots that are indicative of those protein aggregates. However, when we treated the cells with copper ATSM, you can see that the cells don't produce as many protein aggregates compared to no treatment. So we started to also look at different SOD1 mutations. So like I said before, there's over 150 SOD1 mutations, and Natalie Farrell in our lab didn't look at all 150, but she looked at 10 different mutations and treated those cells with copper ATSM, and started to find that copper ATSM was very good at reducing the amount of cells that contained aggregates. So we thought we were onto something here. But then what we started to do was go, well, how is copper ATSM reducing aggregation of SOD1? Well, what we do know is that when we have nearly synthesized SOD1 in a cell, to get it to this step of an intermediate, it requires copper. So what we think is happening is that copper ATSM is delivering its copper component to this nearly synthesized SOD and pushing it into an intermediate state. And therefore it reduces its chances of forming this aggregate. But one thing we started to ask was, well, are there other drugs out there that could potentially help this and push it all the way to the fully folded sod. And so one drug that we were particularly interested in and we helped develop this is work by Dr. Luke McAlary, we found Epsilon was another drug which could help with this pathway. Now, Luke found that what Epsilon does was, rather than work on the first part of this pathway, it actually helps intermediates not get all the way back, get very close to being a fully folded SOD1 protein, and again, reduce the chances that it will go down this aggregation pathway. The last drug that we found as well was telbivadine. Now, this is an antiviral drug. And what we find is that it actually reduces misfolded SOD from going down an aggregation pathway. And that helps the cell get rid of it a little bit better before it can form aggregates. Now, one thing I'd like to mention is that both telbivadine and Epsilon are FDA approved drugs. So we know that they're relatively safe. We kept our search particularly to FDA approved drugs in the hope that that will help get them into clinical trials a lot faster than novel drugs. So the question that we asked was, well, can all three drugs together provide better therapeutic outcomes than just using copper ATSM alone? So to do this, we went back to our cell model. And what we did was either put copper ATSM, Epsilon, or telbivadine onto these cells by themselves, or put them on together. Now, then the first thing we looked at was, well, can these drugs reduce these aggregates from forming? So Luke also developed a very nice and sophisticated pipeline so that we can look at literally millions of cells. So we really get a good understanding of what's going on. Now here we have a cell containing containing a SOD1 mutation with no drugs. And you can see that quite a high proportion of cells contain an aggregate. However, when we treat the cells with either telbivadine, copper ATSM, or Epsilon alone, we see that the percentage of cells containing aggregates reduces quite drastically. So that suggested that we were actually onto something, that by reducing the amount of cells with aggregates, we could be getting better therapeutic outcomes. Furthermore, when we put all three drugs on together, so this is what I call combination therapy, we actually reduced the amount of cells containing aggregates even further. Now that's quite interesting, particularly here, because the concentration of drugs that I used with this combination therapy is actually 8-fold lower than any of these single drugs I've shown alone. And that's quite important, because when you want to introduce drugs for therapy, you want to use the lowest dose possible to avoid any adverse events. So the second thing we asked was, well, okay, yes, we see that there's a reduced number of aggregates within the cells, but what about cell survival? Are we really keeping these cells alive? So you can see here, these cells containing the SOD1 mutation have very low survival rates. However, when we add these three drugs alone, we see that we can start to increase cell survival. And encouragingly, when we added all three drugs together, we drastically increased that number by about 8-fold. So that was quite promising. But what we wanted to do was take this a step further. And like Alison said, cells in a dish are great, but it doesn't replicate the whole organism. There's other cell types that are involved. So what we did was we decided to look in a mouse model. So what this is we've actually got a mouse, and what we can do is introduce a familial ALS SOD1 mutation. And these mice start to develop SOD1 aggregates within their motor neurons, similar to what we see in cells and human postmortem tissue from ALS cases. Similarly to disease, these mice also develop, they start to lose their motor neurons, and they end up with muscle wastage as well. So their muscles start to die and they can't innervate them. And so here I have a picture of our SOD1 mice. And if you gently hold a mouse up by the tail, the mouse will splay its legs. So you can see here that the mouse has spread its legs, and that's typical of any mouse. But as disease starts to progress, and you can see here, this is through the later stages of the mouse's life, its ability to splay its legs becomes less and less until it develops a relatively severe hind leg paralysis. So it's a good model, and it's widely used within the ALS research field. So what we did was, this was work done with Michaela Brown, a research assistant within our lab. We gave the mice either no treatment or a placebo, I guess, copper ATSM alone, and copper ATSM, Epsilon, and telbivadine together, our combination therapy, and asked the question again, well, can all these three drugs together produce a better therapeutic outcome than just copper ATSM alone? So the first thing that we did was we looked at the motor function of these mice. Now, what this rod here in the middle is, it slowly rotates and gets faster and faster, and we can put the mice on this rod, and as disease progresses, the mice will fall off quicker and quicker as their motor function declines. So you can see here that a mouse that is not treated doesn't stay on the rod for very long. But if we give the mice copper ATSM, they're able to stay on that rod a lot longer. Furthermore, when we gave them all three drugs, you can see that we almost double the time that they can stay on that rotating rod, suggesting that we're slowing down disease progression, we're slowing down that motor impairment. The second thing that we looked at was their age of onset. So this is when we first start to observe the mice exhibiting symptoms, so their inability to splay their legs. And you can see that a mouse with no treatment starts to exhibit symptoms around 75 days old. However, mice treated with copper ATSM, it delayed that onset of symptoms. Furthermore, when we give the mice all three drugs together, we see that we're able to delay symptoms, even that onset of symptoms, even further. So this is quite promising. The second thing that we did was look at their weight. So this is our placebo group, if you would like to call it that. And you can see that the mice start to increase in weight, and then the mice start to drop off in weight quite rapidly. And this is a reflection of the fact that the mice start to lose muscle mass as those motor neurons start to die, as their inability to stimulate the muscle causes that muscle to start to waste away. And what we found was that when we give the mice copper ATSM or our combination therapy, so all three drugs, we slow down this decline quite well, suggesting that the mice still are able, or we're slowing down that muscle wastage. And the last thing that we looked at was the survival of these mice. How long did they survive? And you can see that the mice that don't receive any treatment roughly live around 160 days. And we showed that with copper ATSM treatment, we could increase their lifespan by about 10%. Furthermore, when we deliver all three drugs together, we increase that by about 15%. So that, to us, is quite promising, because it suggests that using all three drugs together is better than just using copper ATSM alone. So just to summarize, I think Alison and I both made it quite clear that ALS is associated with protein aggregation, and that protein aggregation disrupts multiple systems that are vital for normal motor neuron function and survival. And that whilst copper ATSM shows therapeutic efficacy in our models, that using Epsilon and telbivadine might provide better therapeutic outcomes. And that we think that this is promising. Whilst we've identified Epsilon and telbivadine, we think we're looking further at potentially what other drugs could reduce protein aggregation as a promising avenue to slow down disease progression. So with that, I'd just like to thank the Yerbry Lab, in particular, Dr. Luke McAlary, Michaela Brown, and Natalie Farrell, who generated a lot of the data that I showed today. In addition to the Donnelly Lab down in Melbourne, and Peter Crouch's lab also at the University of Melbourne. MNDRA, Fight MND and NHMRC for funding our research. So thank you.
Thank you, Jeremy. And thank you to Alison as well. Can everyone hear me better now, by the way? Apparently my microphone wasn't working very well before. That's a fantastic sort of demonstration about how you can sort of delve into the molecular mechanisms of what goes wrong in motor neurons and then actually take it forward to develop potential treatments. One question we have here for Alison. Are there genetic tests at all for SFPQ yet? Is it like you can for SOD1 or FUS or whatever?
Yeah, that's a good question. I think
Not yet is the short answer. We have looked to see if there are genetic mutations in SFPQ that cause MND. And we believe that there are some. There are two that we believe we've identified, but we don't yet have enough evidence to say for sure that they're linked to MND. So it's not screened for at this stage until we can prove for sure that it is causative for the disease. Not yet, no.
So will that be a case of just looking in bigger databases like the Project Mine, etc.?
Yeah, absolutely. Yeah, so we've looked at the Project Mine data. We've gone through all our familial cases, and we have hundreds that we've looked at. And as I say, we have identified mutations that are present in single patients that aren't in people that don't have MND, which makes us think they may well be important. But until we sort of get another person with the same mutation that has MND, we need a bit more evidence, either from a family member who maybe also suffers from it. So yeah, additional data sets. We're always talking to collaborators and getting them to screen SFPQ and other genes that we're interested in as well. So yeah, that is ongoing.
Okay. And since SFPQ, you said, is involved in sort of so many systems, does that mean it's probably not a good target itself to develop drugs from? Is there a risk that you can sort of cause more harm by disrupting it?
Yeah, I think that's a really good question and probably true. Yes. Yeah, it's sort of increasing its expression or reducing its expression is going to affect a lot of different systems throughout the body. So yeah, that's why we're looking for more specific targets, sort of downstream and causes of those changes. So yeah, I think that's probably a very fair assumption.
And a question for Jeremy now. So those three treatments, has anyone else tried them in combination for other diseases where you get that sort of protein aggregation? I mean, you get similar kind of aggregates, different proteins, obviously, though, in, say, dementia with tau. And has that approach been used before?
Not as far as I'm aware. We are looking, or we've been working with collaborators to look particularly at Epsilon. Epsilon seems to work with some other proteins. I don't want to say it works with all, but it definitely seems to show some sort of anti-aggregation properties or helps with protein folding of other proteins. But as far as I'm aware, no one else has looked at those for other diseases.
Okay, cool. And the next question is, so where next for you, Jeremy, with this study then? You've shown some interesting results in mice. A part of that is, are those treatments from birth or from early on? Are they pre-disease presentation, or are they actually treatments after the symptoms have already appeared in the mice?
Yeah, so at the moment, we've only tested it at a pre-symptomatic stage. So we haven't tested it later on down the track. We definitely think that is definitely a worthwhile experiment to do later down the track, to start to look and see whether we can slow disease progression after we already see symptoms existing. Yeah, so look, that is one step. The other step we're doing at the moment is looking to see whether, so riluzole is the current treatment, or Rilutech is the current treatment for ALS, and we're just making sure in the mice that we still see the same effect whilst using riluzole to make sure that there's no adverse effects, so that patients could potentially take both.
And also, I mean, obviously, you're probably aware that tofersen is in the news a lot at the moment. Hopefully, we'll hear in a couple of weeks about whether it's going to be approved or not. But obviously, that is a protein that specifically targets SOD1.
Yeah.
What thoughts do you have about how this might interact with the systems? I mean, does tofersen affect all the systems you were showing us that SOD1 interacts with? Would tofersen affect protein aggregation, or could you treat people with tofersen and your combination therapy? Is that a potential?
So just for anyone out there that doesn't know, tofersen actually, that's right. No, tofersen actually at that RNA level knocks it down so that the cells don't produce as much of that SOD1 protein, which is the protein that starts to aggregate. What we think we know from tofersen, looking at other studies, is that tofersen can't knock down all of SOD1 within the cells. So the cells are still going to produce lower levels, but somewhat A level of that mutant SOD1 protein, which will go on to aggregate. So what we think is that potentially tofersen could be used with our treatment approach, so that tofersen knocks down the protein so that it's not as expressed as highly as it normally would, and that any leaky protein that is being produced, our drug combination could help stop that aggregating.
Cool, that'd be really interesting to see.
Yeah.
Okay, a question back for Alison. This is a more general question, but so when both of you actually, do patients with a familial link tend to have the same genetic mutation? So obviously, families always carry the same exact mutation through the family? And I guess this is even more generic question. How often do people are they aware of the specific mutations they have? That's more of a genetic counselling question, probably. But anyway,
Alison, I think you're the geneticist or closest to the genetics.
I wouldn't go
that
far. Yes, I suppose first part of the question, within the one family, typically it is the same mutation. And that's, I guess, when I was talking before about getting enough evidence to link a mutation to disease, that's the evidence that we need. So multiple family members that have the same mutation leading to disease. So yes, it typically is the same mutation that's passed down the generations. Sorry, what's the second part of the question?
Yeah, and are they generally made aware if they do have a mutation?
Yeah, as you say, there's genetic counselors out there who do an amazing job basically talking families through whether they want to know. So essentially it comes down to some families want to know if they carry a causative mutation, some families don't. So it's very much up to the individual. Yeah.
Yeah, I think the choices there is one, do you want to get tested or not, and two is, do you want to find out the results of those tests?
Yeah, it's a difficult question.
Yeah, and they're decisions that you should always take in consultation with your neurologist or your genetic counsellor as well. They're not to be taken lightly at all.
Absolutely, yeah.
And here we have a bit of a question coming in from Luke McAllery, who we all know, who's a researcher who works with Jeremy down in Wollongong. But he's asking, how different is the SFPQ protein in zebrafish compared to the human? Does it behave the same way?
Yeah, that's fair. I guess similarity wise, it's roughly, the sequence of the protein is roughly 80% the same between the two. So it's not identical, but they do behave the same way in the sense that sort of where they're localized. Less is obviously known about what it does in the fish. So we're assuming it has all the same functions since it's so structurally similar. But yes, I guess less is known in the fish, but that is fair. And that's something we've always got to be aware of, is these species differences can certainly be super important, which is why we never rely on a single model. Anything we sort of see in one model, we always sort of confirm is still relevant in another model. Hopefully that answers the question.
Yeah, and a bit further on that, I mean, what is the sort of, I guess, rate of translation almost from zebrafish, say, to mice? If you see an effect in a zebrafish, do you usually see it in a mouse, or is it a reliable pathway?
I think I want to say yes. I don't have numbers to back that up. We do have a group in the centre at Macquarie who basically they're testing hundreds of drugs in the zebrafish, and drugs that show promise in the fish they then take into mouse models. As I say, the only really promising ones they've found, they've sort of got hundreds that they've tested in the fish down to, I think it's about 4 or 5 that show real great promise in the fish. And they're still in the process of seeing what it does in the mouse model, unfortunately. So I can't sort of answer that with hard facts, I'm afraid. But yes, I mean, I would hope so. Otherwise, a lot of the work is, problematic. Yep.
And speaking of sort of translating models, do you have Jeremy's book, I guess, Jeremy, but also for you, Alison, do you have iPSCs, the human neuron cells set up to at least try your approaches out in those cells?
Yeah, so we definitely work closely with Professor Lizanne Uwe at the University of Wollongong, who does have those cells. I can't comment of late because I know her iPSC lines get bigger and bigger. But last time I spoke to her, she didn't have a patient with the same mutation that we put in the mouse, but she definitely has cells from patients with SOD1 mutations. So that is something we're interested in looking at as well.
And when we're the same at Macquarie, we don't have the setup ourselves, but we also collaborate with Lizanne, who basically generates the cell lines for us. And yeah, she's amazing. Her team is amazing.
Yeah.
Okay, I think that's taken us through pretty much all the questions I think we have here. That's been an awesome way to really explain